I'll talk about the HIPC Coordinating Center update. And the HIPC Coordinating Center does a bunch of things, which are kind of outlined here. And essentially, I'm going to talk about 4 elements. So we provide admin and outreach both to the centers and the scientific community, admin support for the different centers. We provide help or facilitate data submission from the HIPC centers into repositories, specifically primarily import, that is then used within the immune space knowledge base. We are then providing general access to the overall scientific community to that data, which is in a curated, harmonized fashion beyond what is currently would be an import. And then there are activities for data analysis, and actually Steve will present those. So these are the 4 kind of overall topics here that I'm going to present, starting with the admin and outreach. So you guys know all these things. We have the monthly webinars, which actually have been very well received within the centers, where we kind of learn about each other. We have the subcommittees that we learned. So the center here, we are providing just administrative support to kind of like run these subcommittees. Obviously, the work is done in the committees. Much of this is through the HIPC private portal. If you're on the Immune Space website, you can find it, and that's where we have like our shared internal communication. And there's social media and outreach on X/Twitter. And then there's the main website, which is actually our main outreach tool. And these elements that I just mentioned are already incorporated here. So the private portal, you'll find that under support, and others can go there as well and find support. The outreach via social media, HIPC, Twitter/Blue Sky. We have this little box here of the most recent tweets. And then there's the question, what is HIPC? This is always the people asking it. Essentially, if you go to the website, you then get the explanation of what HIPC is actually doing, what the centers are. You can learn more about the centers, and this is what you guys, each center themselves has control over, of what is essentially the base description of what the centers are. But these base description of the grants essentially are not obviously exhaustive. There's more things that are being done. So on the publications, you have all the publications linked to the different centers listed, which is again for the public to learn what we are doing, but also for us. So if you want to see whoever has worked on, I can't read anymore, unfortunately. Yeah, somewhere there is adjuvants. And then you will find all the papers that have been authored by HIPC centers on adjuvants, and you might find someone else. So, and as was mentioned today, we could do this also with computational tools or so. The idea is really to make this as a way how you can learn what others are doing, because obviously the grand summaries are typically not capturing everything. Then this was a suggestion last year from the steering committee and external advisory group. We should be doing more external outreach, and so we are having a symposium at AAI in a few weeks. If you have nothing to do, Hawaii is a nice place to be. Stop by. We also have a booth there where essentially people can stop by and learn more about HIPC. So facilitating data deposition. Again, our primary means is that the data comes from the centers into import. Then we take the data out of import and actually put it into immune space with some additional validation checks, which are more stringent than what immune space asks from everyone, because they have to take data from everybody in the world, pretty much. Not everybody. And so essentially one of the problems was that there was a delay from the data goes into immune space into import until we see it. And we would then say, well, we actually mean that you have to choose one of these possible values for your cell type, say. So now we have a method that we actually can centers submit the data to import, and while it's in their private workspace, they can submit it for validation and then get feedback, and then they could resubmit, or we fix something on the fly with import. And this is now live in testing in the import side. So once you do your submission to import, you have this button here, and then you'll learn about potential issues that might be coming up later. We've also been co-organizing user sessions. Recordings are on the private portal. And in general, if you have questions, please, contact import staff and or the coordinating center via the portal or anything you feel like. So that is the data submission. And now for data access, if you are on the Immune Space website, again, if you go under data, you kind of get like a summary query format here. My eyesight is so bad at this point. Where you can query for different elements of data in import. And here essentially, oh yeah, I should say this. I wanted to do a live demo. And at the NIH here, through the Wi-Fi, everything takes forever to load. please try it out, but please try it out at home. So it took more than 2 minutes or something to load a page like this. So I don't know what the problem is, but from the hotel, it was really fast. So this is now, as I said, we are adding information to the stuff they get from import. So everything that is curation complete, we have added some information to it. And so now this is supposed to be a query for, well, there is a vaccination in the study. And in this case, I put flu mist as an example. And so now you're getting all the studies that we currently have in immune space that have flu mist vaccination. Again, this is not complete, but there's 3 studies which have 3 arms that are relevant. So this is no longer just saying these are the studies, but in these studies, they can have multiple arms. And these are all, they called them here LAIV, which is live attenuated influenza vaccines. And this, in this case, we know because we carry forward, this is flu mist. These are the participants. This is like across all the studies, the ages, ethnicity, etc., in these arms, in these studies where they had gotten flu mist. Then this is the, then the specific events. We have this concept of events within an arm, so such as the vaccination itself. Here, that is what's listed here, but then you have other events like samples that are being gathered, the assays that are being run. So in this case, there's a bunch of ELISAs. There's much more, right? You have like, again, I can't read, 32,000 different things. So if you want to know, has anybody measured after flu mist vaccination IP10 levels with something, you can query for this and get this data back. And then there's signatures. And Steve will talk about that, how we're going to get into essentially the conclusion from somebody who wrote a paper who says whatever IP10 is up would then make it here into the signatures itself and be tied back to the raw data that this was based on. So I mentioned this. So this is, again, like the query for these are the 3 studies that match the flu mist query. If we now look at one of these, you get the information for the total study, which then has this timeline. So, this timeline where you actually see there's 2 arms to the study. They compared Fluarix and flu mist. And so this is the concept here of these arms, where this is the flu mist vaccination I queried for, but there's more participants who got the different ones. So you're seeing, we're breaking down the entire information of the structure, how the study was performed, to then really enable a breakdown and a comparison, then also potentially across studies, where again, you can merge all the data from all the flu mist arms with all the data from all the whatever healthy controls, or you can imagine anything you want, similar to with like people after hospitalization or not, or whatever you want. So what I'm showing you, as I said, is partial. And as we are doing this advanced curation, we're also finding issues that we're trying to address. So not everything is perfect. At this point, last year we had 20 studies loaded. Now we have 91 studies with timelines loaded. This is out of 173 that we have available, out of 183 that we identified to be HIPC related and or signature related that we want to add. So we identified errors and we're trying to fix those. We add the remaining timelines, then we add the signatures. Steve is going to mention that briefly, and then increase coverage of assay types. As you know, lots of the data that we have is stuff like gene expression data, single cell data, etc., which actually is not living in import itself, but is living in resources like SRA-Geo. And this link between this is the sample studied in import, where I can learn about how it was generated from which donor, to then the specific sequencing file. We talked about whatever TCR profiles, etc., is then going to be kind of pre-done for you in a way that you can run these studies automatically for doing something like, with the data standards, or also with the signature projects. These are the other 2 things on the portal. Sorry, I messed up my end here. I just wanted to show like that this is I think at this point, the portal really is getting useful and it's getting used. So we are at I don't know, 5 times or so the average use that we used to have in the last cycle. And I think as we're adding more of this functionality to it, you can now increasingly do things on the immune space portal for these studies that we have in there that really goes beyond what you can do anywhere else. And again, with this kind of functionality of breaking up this data better, we are really wanting to support what Steve is going to talk about, the effort of analyzing the data that comes from all these HIPC centers in a way that really leverages the different parts and makes something that is bigger than the components, right? Because that's the reason for why we don't have a bunch of R01s, but one network. So take it.
All right, so I'm going to talk about the part of the coordinating center that's focused on data analysis resources and infrastructure. And really our goal here, like Bjorn said, is to support the immune signatures kinds of analyses, these kinds of meta-analysis that Matt talked about, as well as other kinds of resources that are going to be broadly useful across the HIPC centers and across the wider scientific community. There are three efforts within the coordinating center that fall under this data analysis resource and infrastructure banner. And I'll go into each of those three, just a couple slides on each one. But the first is to capture and disseminate public signatures that we as HIPC members publish. The second is to develop and make available a set of multi-omics modules to support enrichment types of analyses. And the third is to support the identification of immune signatures related to the outcomes of vaccinations and infections. And what I mean by that is really the IOF signatures project that Matt just talked about. So first, I want to talk about the capture and dissemination of published immune signatures. So I think over the past several years, we've done a really good job at capturing all of the underlying raw data that a lot of the HIPC centers have been generating, both in import and through immune space. What we recognized last cycle and started to put an effort into is we also wanted to capture the results of the analysis of those underlying data. So in the HIPC cycle 2, we developed a data model for expressing an immune signature that is the results of the analysis that essentially tells you that, okay, genes A, B, and C go up after infection, or protein Y decreased levels are associated with poor vaccination response. We created a data model for capturing that kind of information. And then we want to curate the papers that we are publishing in order to put that kind of information in a machine readable format so that we can all use that as part of our studies. So where we're up to now is the signatures are now fully integrated onto immune space. So when we capture these signatures and make them available in a machine readable format, as Bjorn showed you, there's a tab within immune space now that you can point to, and you can find curated signatures that are associated with the different studies within immune space. As in terms of the coordinating center, we are focused specifically on capturing signatures that are published by HIPC centers. And in that, where we are with that, there's 84 HIPC papers that we've identified and screened for potential immune signatures. 28 we identified as meeting our priority inclusion. We're focused initially on signatures that involve genes, proteins, and metabolites, and as well as the signatures where the data is already available within import. And there's 17 now that we're currently working on and incorporating into immune space. 17 papers, and each of those papers can have multiple signatures associated with each paper. And just one update on kind of what's changed since the last time we presented this. First of all, one thing that's changed is we are now doing this curation primarily within the coordinating center. So last year, we've been trying to work directly with each of the HIPC centers and to get the the authors within each HIPC center to curate their own data and submit through these web-based templates. That wasn't working so well, so we now are actually doing the initial curation in-house, capturing the data, and then we are many of you may have gotten emails from us. We are reaching out to you guys, the authors of those papers, where we need clarification of anything to do with the signatures. But we're doing the initial work now. And secondarily, we've extended the data model for the signatures to capture more data. And in particular, the one thing I want to highlight is this timeline information. So Bjorn showed a picture of the timeline where now within immune space, you can get this graphical picture of the experimental design of the HIPC studies. You can actually follow the arms, any treatments that were given to those different arms in terms of vaccinations or other kinds of treatments that may have taken effect. And now we can link the signature up with those timelines. So for example, an example is in this study of a COVID vaccine response, there was a signature that was discovered within CD14 positive monocytes in terms of the increased and decreased expression of different genes. And that signature will now link specifically to the timeline. So you'll be able to see that this signature relates the profiling of this specimen that was taken 22 days post-vaccination with this specimen here on the time that was taken 1 day post-vaccination. And so there's a direct linkage now between the signatures and those experimental designs and the data that's in import. Okay. Second effort within the coordinating center is the development of multi-omics modules. So as part of HIPC, many of you may be familiar, and we've certainly made heavy use of gene set modules, things like the blood transcriptional modules that came out of Bali's group, or the blood gen modules that Matt was involved in. But over the past years, many HIPC centers have been generating multi-omics types of data. And one of our efforts has been to extend the concept of a module to not be a single omics type, to not be just a gene module or a proteomics module, but to develop this concept of a multi-omics module. So a module that encompasses both genes and proteins and metabolites all together. If you go onto the Immune Space website under resources, you can see the beta version. You can get access to the beta version of the modules as they exist now. And we've put together a pipeline where we can take data from multiple participants that have this multi-omics types of data. Again, our initial focus is on genes, proteins, and metabolites. And we have a pipeline where we do a dimensionality reduction of those data. Current pipeline uses a technique called MOFA. We get these multi-omics types of factors to reduce the dimension, and then we use a feature selection technique based on Lasso to find a minimal set of genes and metabolites and proteins that can account for the behavior of those modules. Version 1 of these modules, which is the one we have available on the website now, was built on a cohort, the cohort that Matt had mentioned from the Impact Network that encompasses over 1,100 COVID-19 patients. This is a really large multi-omics data set, and that's where we developed our version 1, and we're now extending that out to other data sets. The first version covers 16 transcriptional modules, 22 proteomics, 12 metabolomics modules, covering a diverse range of immune processes. We've started to do some comparisons of these modules with the different sets that are out there. So, for example, the heat map on the right here shows the BTMs across the rows, along with the transcriptional module across the columns. And you can see at the resolution that we're able to get now, each of these larger modules actually encompasses multiple different functions represented in the BTMs. And we've now taken an effort to relate these modules to one another. Broadly speaking, some of what we found in these set of modules is that the proteomics and the metabolomics modules are highly predictive of each other. And the transcriptomics modules, for the most part, seem to provide orthogonal independent types of information. And finally, the final effort that I wanted to cover in this part of the coordinating center is the support for the HIPC IOF signatures projects. As was already mentioned, one of our roles is to help coordinate and support the submission of these IOFs, which has been happening for all the years of the HIPC so far. And as Matt mentioned, we now have a signatures IOF and a genetics IOF that were both approved. We continue to maintain the code and access to the previous HIPC signatures projects data on immune space. We've done some training across HIPC for people who want to continue to use that code base. A big effort over the past year was to create the version 2 of the immune signatures data resource that Matt mentioned. We had the first version, which covered 24 vaccines and about 1,400 participants. What we're thinking of for the release of version 2, where the publication is going to be, we're going to be focusing for this publication on the vaccine response, and it's going to cover mainly the bulk data sets. And so what this is going to look like, it includes 100 cohorts from about 50 studies, about 2,400 participants covering 34 different vaccines, almost 8,500 samples. So all of that data is actually now in immune space. So it covers many HIPC studies, but we also did a literature search and we found many non-HIPC studies as well. And we went through the effort now of pulling in those non-HIPC studies. All of those non-HIPC studies are also now in import. We've connected those up. We have those all processed through a common pipeline. And so you'll be able to get access to this data set of 100 different cohorts, again, covering 34 different vaccines and almost 8,500 expression samples. And that's what those signatures IOF projects are based on. And that's going to be available to you and the wider community as well. Where we see this going next, as was mentioned, is right now that resource is based mainly on bulk data. But many of the HIPC centers now are focused a lot on single-cell RNA-seq plus B-cell receptor, T-cell receptor, site-seq, and so forth. And so that's where our focus is now, on extending the resource into those single cell data sets. First thing that involves is standardization, as was mentioned by both Matt and Thomas. We have been working for the past year or so on a new HIPC standard for single cell RNA-seq that includes both B cell receptor and T cell receptor data. We've gone through the process of drafting it. It's been seen by the representatives of all the HIPC centers. It's been presented at the bioinformatics subcommittee and annual meetings, and it's at the stage now where we've got the draft that is at the steering committee, and maybe we might approve it this afternoon, or it may, depending on timing, may have to wait until the next virtual call. But that's now waiting to be approved by the steering committee. And that is, for anyone who hasn't seen it yet, it should have been seen at least by the bioinformatics committees and the representatives. That is also on the on the immune space website in the discussion area. And finally, as was mentioned, also in addition to providing the resource and the standards to move to the next version of the resource, we are also supporting the development of those pipelines by carrying out various benchmarking studies to make sure when we make available those data sets for the Signatures project for the wider community, we are using the best methods that we can. So in particular, we've been benchmarking integration methods. I think this was a question that was asked online. So we did do a comparison of multiple different state-of-the-art integration methods, where we did find, for example, that of the methods that were published at the time, Harmony was the best performer, but some of the observations here led Li-Ying and her team to develop a newer method, SC-Craft. But again, we're carrying out those kinds of benchmarking studies. So when we make available those data sets, we do have some underlying reasons for why we made the choices that we did at each step of the analysis pipeline. And finally, I wanted to just kind of leave things just with a sort of a call for interest. One of the things that we see as our next step in terms of, as we think about how do we make available an immune signatures data resource that involves single cell data, integration is sort of one challenge that we presented, but cell type annotation is another challenge. And many different groups have kind of brought this up at past annual meetings, sort of at different points in this process. But we see this as sort of the next challenge that we as a consortium and the coordinating center is gonna have to face. How do we sort of annotate these studies across our centers in a common way? And we think there's an opportunity for some collective effort where we could generate some reference atlases that are annotated in a uniform way across the center. So we'd like to, for example, provide some standardized cell lineage trees, for example, based on the cell ontology, where each of the centers could be involved in annotating their own data sets, but using a common terminology that we could then provide as a resource, and we can all annotate ourselves in a common way. And I will leave it at that, just to say that if you're interested in this kind of effort, please reach out to me or Bjorn or to Li-Ying. All right, and with that, I'll invite Bjorn back up here as well in case there are questions. But just to say that this has been an effort of multiple people listed up here, several of whom are out in the audience today, several more of whom weren't able to make it in person. Again, multiple people involved, both at the coordinating center as well as NIH, and the import team have all been very involved in this. And the slide I will leave up at the end are the QR codes, in particular, social media on Blue Sky. We have a new Blue Sky account. And if you have not followed us yet or gotten on Blue Sky, I really encourage you to link up with us and follow us. If you have a public HIPC related publication and you sort of tag us on Blue Sky, we will like and repost and whatnot.